This is Project 4, Humoral Immunity by Kylie Hernandez, Rowan Randolph, and George Escobar. This is just a slide listing all of our roles.
So the B cell immune response goes through a variety of phases. First phase is antigen recognition, where the cell recognizes an antigen on the microbe. Then it goes, and then it activates and goes through proliferation, where the cell multiplies, and differentiation, where the BCR differentiates. And then after this, it'll produce effector cells such as plasma cells and memory cells. Now, there's two different types of antibody responses. You have T-dependent and T-independent. Now, T-dependent, what it is when the B cell recognizes a protein antigen, and in order for it to do that, it needs a T helper cell. And usually, what this makes is it makes stronger effector cell functions, like memory B cells, isotype switched antibodies, stuff like that. And then next is T-independent, where instead of taking a protein antigen, it takes any other antigen that's not protein, like polysaccharide. And it doesn't need a T helper cell, but the downside is that the effector functions of the effector cells are going to be weaker. Here, look, weaker effector cell functions. So, innate immune signals can also activate B cells. For example, you have the complement protein C3d, which can bind to a microbe, and then it will bind to the receptor on a B cell called the CR2, and they will basically act as more or less kind of like a secondary signal to activate the B cell immune response. The TLRs can also recognize PAMPs or anything related to microbes.
Heavy chain isotype switching is a T-dependent response in which naive B cells, initially all IgM or IgD, receive the necessary signals from the CD40 ligand on follicular helper T cells to switch their Fc region. The class immunoglobulin switch here depends on the cytokines produced. For instance, interferon gamma signals the B cells to switch to the IgG class. Interleukin-4 tells B cells to switch to IgE, and transforming growth factor beta tell them to switch to IgA. Affinity maturation occurs through a process called somatic hypermutation, a process in which there are point mutations introduced to the variable region of B cells, creating a pool of different affinity B cells, some high and some low. Eventually, only the B cells with the highest affinity receptors for a specific antigen will be selected to survive as they continue to be produced. These new B cells are now selected so that only the highest affinity B cells will survive, as the high affinity B cells are able to grab onto the antigen carried by follicular dendritic cells easier. They are then given signals from follicular helper T cells to survive or become memory cells. Antibody feedback occurs in order to prevent overactivation of the humoral response that could potentially lead to tissue damage. At the same time, a B cell's BCR recognizes an antigen, an inhibitory receptor, Fc gamma R2b, binds to the Fc region. This Fc receptor contains immunoreceptor tyrosine-based inhibition motif on its tail, which sends signals to the B cell that block the further activation of the B cell.
Effector functions of antibodies. There are 5 main effector functions. Firstly, with neutralization of microbes and microbial toxins. Antibodies bind to microbes or toxins and block their ability to infect cells. And second, opsonization of phagocytosis. Antibodies coat microbes and phagocytes recognize Fc portions via the Fc receptors and enhance ingestion. Then there's antibody-dependent cellular cytotoxicity. Natural killer cells bind to antibody-coated cells via the Fc receptors, and this leads to the killing of infected cells. And then there's eosinophil/mast cell-mediated reactions where IgE will bind to parasites and triggers degranulation, which leads to the parasite destruction. And then there's activation of the complement system, mainly through the IgM and IgG, which activate the classical complement pathway. As can be seen here in the image, how that B cell produces those antibodies, and this can lead to the neutralization, the opsonization, and then the NK killer cell causing apoptosis. And then those antibodies can also cause complement activation, and this leads to the lysis of microbes, the phagocytosis of microbes, and that inflammation. Mucosal and neonatal immunity. With mucosal immunity, it protects mucosal surfaces such as with the GI and respiratory tracts, and it functions mainly to neutralize pathogens and mucus and prevent microbial attachment to epithelial cells. So as can be seen here, how the Ig producing plasma cell produces that geometric IgA, and the poly Ig receptor binds to that IgA, and it leads to an endocytosed complex of IgA and poly Ig, and then it is secreted. And with neonatal immunity, it provides passive protection to newborns. Before birth, it is done through the placenta with IgG crossing the placenta via Fc receptors, and it protects the fetus before birth. And then after birth, it is done with breast milk, which is rich in IgA and protects mucosal surfaces of the infant, the gut especially. Evasion of humoral immunity by microbes. So microorganisms have evolved strategies to escape antibody-mediated defenses. This can be seen here with the antigenetic variation as it's changing those surface antigens, and then the inhibition of complement activation as the microbes inhibit complement activation or MAC formation, and then there's the blocking by hyaluronic acid capsule as capsules prevent opsonization. And with vaccination, it is a process of stimulating protective adaptive immune responses against microbes by exposure to non-pathogenic forms. It induces humoral immunity by generating memory B cells and antibodies. And as can be seen here, just the many different types of vaccines that have been produced in the modern day. Here are references. Some images were created with BioRender. And thank you so much for your time.